In Essential Task A2, we are going to describe the electrical process of neural firing and the chemical process of transmitting a signal between neurons. In Essential Task 8.1, we diagrammed the neuron and denoted specific functions related to regions in the neuron. Today, we are going to add the second element to our conversation. We are going to talk about what happens when one neuron communicates with another. There are 7 total boxes in today's concept map. The large heading at the top is neural communication. Then we're going to divide that up into electrical, which has 4 total boxes, and then chemical, which has 3 total boxes. So the 7 boxes that you see currently on the screen is the entirety of today's concept map, which is why I recommend a horizontal piece of paper. I'll return, though, to the task text, allow you to copy down the portion in yellow before proceeding with the video. So we will start all the way on the left and talk about the electrical steps in neural communication. This is going to be one of those essential tasks where, as we go box to box, the big picture might not make sense until we zoom back out and synthesize all 7 of these boxes in combination with one another. So I'll totally understand if we're going from box to box and the information doesn't make a ton of sense until we actually get to the very end. When we talk about the way a neuron communicates with other neurons, we need to denote a couple different terms. The first term is what we call resting potential. If you, again, see this from a far out, zoomed out look, you'll notice I have the number 1 next to this. I'm sort of going to proceed from step 1 to 2 to 3 and talk about the sequence by which we see a neuron shift from what's called resting potential eventually to what's called action potential. But first, we'll start with resting potential. Resting potential is what we call a neuron that is not currently transmitting or receiving a message. It is, as the name suggests, resting. It is not being activated. It is not being stimulated. Those sensory neurons in certain parts of your body that are not being actively stimulated through the sense of touch are currently in resting potential. Why is a neuron called resting potential? Well, when a neuron is just resting, the interior inside of the neuron is relatively more negatively charged than the outside exterior environment. So if you take a zoomed in look at a neuron when it is resting, as I have done here at this diagram, this is a diagram zoomed in on the axon, specifically the space between 2 myelin sheath. So I'm looking right here. It actually has a formal name by which we don't need to worry about rods of Nanvier, but we look at this little region here of the axon. You'll notice I have on the interior of the neuron a bunch of negative signs, and on the exterior of the neuron I have a bunch of positive signs. This somewhat simplifies the actual chemistry going on here. It's a little bit beyond the scope of AP Psych. If you took AP Bio, you might know a little bit more in depth exactly what's going on here. But for the circumstances that we're in, for the context of AP Psych, what we need to know is that when a neuron is just resting, not transmitting a message, not communicating with other neurons, it is relatively more negative on the interior, at least relative to the outside environment. So you'll notice I put a lot of negative charges. There are negative ions on the interior of the neuron, inside of the neuron, that makes it negative. As a mnemonic, when I think of just the word neuron, I know negative and neutral can in and of themselves both be connected to that NE. I really tried to emphasize, though, neurons are normally negative. I think that flows quite well, that neurons are normally negative. And what you're going to see is that when a neuron goes from just resting into actually transmitting a message, there's going to be a shift in its charge. And that shift in charge is going to be a big deal when talking about how neurons communicate with one another. So we'll start off resting potential. Neurons are not communicating with other neurons, not transmitting messages. Why is it classified as resting? The interior is more negative, at least relative to the exterior. That obviously is going to change. So now we're going to shift from 1 to 2, and we're going to talk about what happens to a neuron when it is in fact stimulated, when it is in fact communicating with other neurons. When that occurs, we're no longer going to use the term resting potential. Instead, we're going to use the word action potential or neural impulse. Those two terms are synonyms of each other. They describe the same process. So action potential or neural impulse, it's the same thing. Each of these terms references a literal brief electrical charge that occurs after a neuron is sufficiently stimulated or fires. You might hear the phrase neuron firing in this unit. That denotes the change from resting to action potential. Well, why does a neuron change from resting to action potential? Well, when the receptors, which again, on your diagram from Essential Task 8.1, was the surface of the dendrite, so the furthest to the left in your diagram, when those receptor sites are stimulated or activated, that stimulation causes positive sodium ions on the exterior outside of the neuron to rush through the membrane and into the neuron itself. When there is this big rush of positive ions into the interior of the neuron, it briefly causes a change in charge, at least relative to the environment. And it literally causes an electrical charge to be produced. That individual electrical charge itself is called an action potential. And it doesn't just stay at one part of the neuron. It literally travels across the neuron. Back in Essential Task 8.1, I gave you the mnemonic DAT way. It denoted the order of dendrite, axon, and terminal. It also tells you the direction that action potential travels. Action potential literally moves that way through the neuron, dendrite, axon, terminal to the very end. So in conclusion, when a neuron is in resting potential and it is stimulated at its receptors on the dendrites, the furthest to the left of your diagram, that neuron, which is normally negative, suddenly flips to positive, at least relative to the environment. It produces a brief electrical charge. We call that an action potential. And that action potential, that electrical charge, literally travels across the neuron. So if you look at the diagram at the bottom left, notice I have some of these positive, they're technically sodium ions. Notice how I now have them not only on the exterior, but I have some also on the interior. That essentially flips the charge of the neuron. If you've ever looked at what a neuron looks like when it's active, you'll actually see diagrams like this. This isn't a real diagram of a real neuron, but sort of an artistic evaluation or depiction. You'll actually see this little charge. It's not there by accident. It's literally an action potential. You can literally see it travel across the neuron. There are a couple terms relative to the electrical aspect of neural communication that we need to talk about. The first is to acknowledge what's called the threshold of excitation. This is the quantity of stimulation necessary to cause a neuron to shift from resting to action potential. If the stimulation at the receptor sites is not great, it's a minor stimulation, it may not be sufficient enough to trigger that neuron's shift from resting to action potential. There is a minimal amount of stimulation necessary for a neuron to go from just resting into action potential. And it works a lot like a toilet flush. Really emphasize that toilet comparison. When you flush a toilet and you pull the lever down, it doesn't matter if you put the minimum amount of pressure to produce the flush, or if you hit it as hard as you can, each flush is going to have the same level of intensity. Flush is the same every time. Neurons work in the same capacity. We call this the all or none law, meaning the second the neuron has reached the threshold and shifts from resting to action potential, that sodium is going to rush on the inside. It's going to produce an action potential, and that single action potential proceeds that way across the neuron the same way every single time. There's no higher intensity action potentials. Each action potential works quite similar to the way dominoes work or the same way a toilet flushing works. The terms here is threshold, the minimal amount necessary to trigger resting into action potential, and then denoting the all or none law, that each action potential has the same intensity as the previous action potential. Once a neuron has shifted from resting to action potential, there is a brief window of time called the refractory period. This is the last box on the left region, denoting the electrical steps in neural communication. So we're proceeding to the box here. The refractory period is a brief period of time, essentially where the neuron needs to pump out all of the sodium that rushed in so that it can return back to its equilibrium point, which for a neuron would be a relatively negative state, at least on the interior. So there is a short window of time after a neuron has triggered action potential where it cannot be stimulated again. It needs to pump out the sodium. It needs to reset itself. It needs to sort of repolarize itself, to use some terminology. That's called the refractory period. I like to call this the reset period. It's the small window of time where the neuron resets itself back to resting potential, where it would have a relatively negative interior, at least in relation to the exterior. That's the electrical component. It basically gets boiled down to a neuron's just hanging out in resting potential. It is stimulated. It flips from negative to positive for a moment, literally causes an electrical charge that travels that way across the neuron. Once that action potential, though, gets to the very end of the neuron, we're now going to add the chemical element to neural communication. Do note that there is a third box down here in the corner you might not be able to see because of my camera. So now we're going to shift to the chemical side. We had a neuron that was resting, it shifted to action potential, and now we're going to add the chemical component to it. Here is where we're going to reference those vital chemicals known as neurotransmitters that I alluded to in the previous essential task. This is also where we're going to start to talk about the fact that one neuron isn't just in isolation. Neurons communicate with other neurons, and it is here that we're going to talk about that. First, we're going to talk about what's called the synapse, or the synaptic cleft, or in some cases, the synaptic gap. When you hear the term synapse, it technically references the region between two neurons, the very end of one neuron and the beginning of the next neuron. As we talked about with the diagram, the end of one neuron is the terminal. We call it the terminal buttons. The beginning of the next neuron would start with the receptors on the dendrites. So the space between the terminal on one and the dendrites on another is called the synapse, that actual literal space. Neurons do not actually touch one another in order to communicate. So they are very, very close to one another. But nevertheless, that region there is called the synapse. Occasionally, you might hear or see a question that asks you to precisely denote just the space, not necessarily the presence of each neuron. The space can sometimes be called the synaptic cleft or synaptic gap. But nevertheless, whether we talk about the synapse, the synaptic cleft, or gap, we're essentially talking about this same general region, as denoted here in the diagram. The space between the end of one neuron and the beginning of the other. Often we'll use terminology such as the presynaptic and the postsynaptic neuron. The presynaptic neuron means the neuron before the synapse. Again, the synapse is the space between the two. So the presynaptic neuron would include the terminal of the first neuron, but then also the synapse includes the dendrites of the postsynaptic neuron, post meaning after the space. So the synapse is essentially a region between two neurons, and it very simply contains the end of one neuron, so the T in that way, the terminal buttons, which contain the synaptic vesicles, in actual small space, whether we call that the synaptic gap or the synaptic cleft, and then the beginning of the next neuron, the postsynaptic neuron, and the receptors on the dendrites of the next neuron. If you want to use the dat-way mnemonic to envision this, think about the word dat being spelled twice back to back. The synapse would be the location between the T and the D, that actual space between those two. So if you look here at the diagram, this here at the top denotes the presynaptic neuron. The one at the bottom denotes the postsynaptic neuron. And then this actual space between it denotes the synaptic gap. So the synapse is the entire region depicted here in this current diagram. And it is here where neurotransmitters will take effect. And we'll talk a little bit about that in the next box. As a mnemonic, when I see the word synapse, I see the word nap in the middle, and I think about the idea of literally laying a hammock between the T and the D in the words dat. Again, there is a literal space between each neuron in the synapse, just as there is a space between the letters T and D. So obviously you wouldn't actually do this, but I kind of envision a hammock being laid out between the two. Throughout this unit, we've defined briefly, or alluded to rather, neurotransmitters, but here we will definitively define neurotransmitters, and in a later essential task, we'll denote the different types of neurotransmitters in your body. But neurotransmitters, as we established in the last essential task, when a neuron is resting, hang out in the synaptic vesicles at the end of the axon terminal, in the terminal button. So if you look at this diagram, this top region here is the end of a neuron. So you'll notice in these little pods, they look like little triangles, but those are neurotransmitters. When a neuron goes from resting to action potential, it causes these little chemicals, known as neurotransmitters, to exit out of the neuron, literally exit and leave the neuron. Those neurotransmitters go into the synapse, or the synaptic gap, the literal space, and it is these chemicals that bind and stimulate the receptor sites on the next neuron. So as the action potential travels that way across the neuron, when that brief electrical charge, again, caused by the rush of positive sodium ions into the neuron, reaches the end of the neuron in the terminal buttons, it causes the synaptic vesicles, these little pods, to release the neurotransmitters that they contain into the synapse, as you see here in this diagram, and it is those neurotransmitters that stimulate the subsequent postsynaptic neuron. After that, those neurotransmitters are reabsorbed back into the neuron that sent them during a process known as reuptake. What that essentially means is when your neurons release neurotransmitters like serotonin and dopamine and glutamate and all of the others, endorphins, those neurotransmitters are released out of the neuron. They trigger and stimulate the next neuron. And then after they've done that job, they get reabsorbed back into the neuron that originally sent them, meaning you technically reuse the same dopamine. You reuse the same acetylcholine, the same endorphins. Those chemicals in your body are reused every time those neurons are activated. And then they get reabsorbed back into the neuron that originally sent them, and they wait there until the next time that neuron is used.